Okay. I think that's going to work. All right. So y'all might remember last week, maybe even the week before, whenever I posted the lesson on Thomas Hunt Morgan, right at the end of that lesson, I talked a little bit about linked genes. And this was something that Morgan discussed in his research, the idea that if you have genes for different traits, in this case, a gene for body color, talking about fruit flies here, so body color, and then a gene for wing shape. If those two genes just happen to be physically located on the same chromosome, we consider them to be linked. And by calling them linked, what we're really saying is that those two genes are not going to fully assort independently. They're not going to follow Mendel's law of independent assortment the way genes really ought to do. And Morgan was really the first to realize this, that when genes are linked, they don't follow Mendel's law of independent assortment, at least not always. And he discovered that in fruit flies, the wild type body color, this would be the B plus allele, codes for kind of a grayish color body. That's what color you see when you look at most fruit flies. But a small percentage of fruit flies have a black body mutation. The body color is much, much darker. And that turns out to be a recessive mutation, a recessive allele. And so this would be a fly that is heterozygous. It has gray body color, but it's also carrying the recessive black body allele. And then, of course, normal wings, VG plus, is dominant, and then vestigial wings is recessive. So Morgan discovered that these two genes are, in fact, linked. They're located physically on the same chromosome. But in the early 1900s, and really up until probably up until about 20 years ago, 30 years ago, you couldn't physically look at a chromosome and see the locations of the genes. Nowadays, we have ways to make genes light up and glow, and you can actually see exactly where they're sitting on the chromosome. But in T.H. Morgan's time, this was quite impossible. All he could say for sure is these two genes are located somewhere on the same chromosome, but he couldn't see where they were sitting on the chromosome. But he realized something really important, that when crossing over happens, when this chromosome and this chromosome come together as a pair in prophase 1 during sperm making or egg making, crossing over happens, as you know, which means a little piece of the blue chromosome is going to break off, a little piece of the red chromosome is going to break off, and remember, they're going to switch places. Crossing over is swapping DNA back and forth between homologous chromosomes. And he realized that when that happens, there has to be a physical break. This chromosome, let's say, breaks right there. The other chromosome is going to break in the corresponding location. And so that's how this little piece and this little piece can switch places. And where that break occurs is completely random. And it's different every time, and it's totally random. It could be anywhere along that chromosome. So he said, if I could calculate how often that break occurs in between two genes, it'll tell me something about how far apart they are. Genes that are linked can be far apart, like this, or they can be really close together. And remember, he couldn't see their location, so how do you know? Well, that place where that break happens is completely random. It could be here, it could be here, it could be here, it could be here, it could be anywhere. So the likelihood that that random break would happen in between the two genes, if they're really close together, it's pretty unlikely that the break would happen in between the two genes. But if they're far apart, there's pretty good odds that that break is going to happen somewhere in between them. And Morgan figured out how to calculate that. He learned how to calculate mathematically, and I'm going to show you how to do it, how often, how frequently that crossover event occurs in between the two genes. And that gives you an indirect way of sort of figuring out, at least relatively speaking, how far apart the genes are. So what you're going to calculate is called percent recombinance, which means what percentage of the time is there a crossover event in between the two genes. If that number is high, if you get a high number, a high percent, that just simply indicates that the genes are pretty far apart. But on the other hand, if you get a low percent, it means that those genes are relatively close together. And by calculating this, you can start to get a sense of where the genes are physically located, and you can map them out. These were the first primitive gene maps that were devised. Again, this is in the early 1920s. We have much more sophisticated technology for doing this these days. But this is a basic mathematical approach to mapping the locations of genes on chromosomes. So with that in mind, let's look at this ugly handout that I gave you. And let me show you how to make some of these calculations. And I'm hoping this will show up okay for you folks that are watching the recording. I just don't know how to make that any better. Okay, so if you look, this is a Punnett square, or it's going to be a Punnett square. I'm crossing a gray body normal wing parent with a black body vestigial wing parent. And I've even given you the genotypes of the two parents. So the gray body parent is actually heterozygous, B plus B. It's got a gray body allele and a black body allele. And it's also heterozygous for wings, normal wing and vestigial wing. So heterozygous for both. And it's been crossed with a fruit fly that is homozygous recessive for both, BB for black body, and VG for vestigial wings. So the B allele, just so you'll remember this when you look back, the B allele codes for black body, and the VG allele codes for vestigial wings. And whenever you do a cross like this where one parent is homozygous recessive, this is called a test cross. So sometimes you'll see that reference when somebody does a test cross. That means they cross some parent with a homozygous recessive parent, which is what we've done here. So this is a dihybrid cross because it's two traits, body color and wing shape. And so it would be a 16-box Punnett square if we did the whole thing. But you could actually take a pretty nice shortcut on this one because this parent right here can only make one type of gamete, right? It has to pass down B, and it has to pass down VG. That's all it has. So that's the only kind of gamete it can make. But the other parent can make 4 different gametes, right? Because it can pass down B plus VG plus, or B plus VG, or BVG plus. Don't you just hate this system for writing genes? And then BVG. So this parent can make 1, 2, 3, 4 different gametes. Therefore, there are 4 different types of offspring that are possible. So we could get this one, B plus B, VG plus VG. What would that fruit fly look like? Gray body, normal wing. All right. Or we could get gray body, vestigial wing. Or black body, normal wing, and finally, we have the possibility of black body vestigial wing. So 4 phenotypes. And according to the Punnett square, it should be 1/4, 1/4, 1/4, 1/4. That's what the Punnett square predicts. That is our predicted, or I could have said expected outcome, right? It should be 1/4 like this, 1/4 like this, 1/4 like this, and 1/4 like this. Now, look at the numbers. These are T.H. Morgan's actual data here. Did he get 1/4, 1/4, 1/4, 1/4? No. We don't even have to do a chi-square on this. Obviously, our observed, which is these numbers, is way different from expected. There's not even a point in doing chi-square. It's obviously way off. And Morgan was smart enough to realize a couple of things. First of all, he was smart enough to realize that the reason it's this far off, the reason we have way more of these and not as many of these is because the two genes are linked. They're not obeying Mendel's law of independent assortment. Therefore, the predicted outcome from the Punnett square is bogus. It's not matching the actual result. It's because these genes are linked. The body color gene and the wing gene apparently are physically on the same chromosome. He also noticed something else, and you can notice this too. When I look at my two big numbers, the 985 and the 993, and I look at those genotypes, this one and this one, compare that to the parents. 985 of these flies look exactly like this parent. You see that? And 993 of them look exactly like who? The other parent. So those are called parental types. Morgan said those are the parental types. But the other ones don't look exactly like either parent. Neither one of these parents was gray body vestigial wing, and neither one of these parents was black body normal wing. So these ones, these offspring, according to Morgan, we call these recombinant types. They're the ones that are not parental types. And by the way, it'll always be the two smallest numbers. It will not always be the two middle numbers. That's just the way I set this up, but it will always be the two smallest numbers. And here's what that means, because I know this is getting a little confusing. Recombinant types are the fly babies that resulted from a crossover event that occurred in between the two genes. So if we can calculate what percentage of the offspring are like this, that'll tell us how often, how frequently a crossover event occurs in between the two genes. Okay. Now, calculating that is super easy. The first thing I need to know is the total number of fly babies. So add those up, straight across, and check me on this. I got 2,300. Okay, so there were 2,300 baby flies, of which 322, I just added these two smallest numbers together, 322 of them were recombinant types. Well, I can calculate that as a percentage. If I want to know what percentage of the offspring are not parental types, that's easy. That's 322 divided by 2,300, which is going to give me 0.14. Check me on that as well. If I want that to be a percent, how do I do that? Move the decimal two places, multiply by 100, so it works out to be 14%. So if I'm trying to get an idea of how far apart these two genes are, they're sitting there on the same chromosome, and I want to know whether they're far apart or close together, well, I kind of have at least a relative idea that 14% of the time, this random event is happening in between the genes. If they were further apart, that number would go up. It would be a higher percentage. So What Morgan did is he said, let's take that percentage and let's refer to that as a map. Each percentage is a map unit. So if we're drawing a map, I'm going to draw just a horizontal representation here of a chromatid, which is 1/2 of a chromosome. If I draw a chromatid and I'm trying to map the locations of the genes, I can't use actual distances like millimeters or micrometers because chromatids come in different lengths. Some chromatids are really small, some are really big. But I can get an idea of the relative distance between the body color gene and the wing gene. I now know, based on my calculation, that the distance between those two genes is 14 map units, 14 MUs, or map units. That does not correspond to an actual distance. It's just a relative indication. And all I did to calculate that, and I know it's easy to get lost in the details here, but all I did was I took the four numbers that were given me, I added the two smallest ones together, and I divided by the total. That's all I did. And that tells me, relatively speaking, how far apart these genes are. Now, it gets more interesting if you throw a third gene into the mix. Turns out that the body color gene and the wing gene are on the same chromosome as a third gene called the cinnabar eye gene. It's a weird eye color mutation. As you know, most fruit flies have bright red eyes, but some fruit flies have kind of a cinnamony brownish red color called cinnabar. And if you turn over to the second page, you'll see some data that will reveal to us the distance between the body color gene and this new cinnabar eye gene. So by the way, CN stands for, I think I'm spelling this right, cinnabar eyes. Okay. This is going to tell us the distance between the eye color gene and the body color gene. And I'll save you some time here. Now that you understand the concept, you don't need to do the Punnett square every time. I only wanted to do that on the first one so you could see what was going on. Now that you know what you're doing, just go straight to the data. Which two numbers are we interested in? The two smallest ones. Why are we interested in the smallest ones? Because those are going to be my recombinant types. Those are the ones that are going to tell me where crossing over has occurred. So I'm going to add those together. And I got 423. And the other thing I need to know is my total number of fly babies, which in this case was 2350. So I added them all up. And then I figured out how many recombinant types I had. It's always going to be your two smallest numbers added together. 423 divided by 2350 times 100. Check me on this, I got 18%. Everybody with me? Okay. Now, it's like putting together a puzzle. Now we can sketch two possible maps. Now, one of these is going to end up being the correct map, and one is going to be the wrong map. But there are, at this point, based on what we know, there are two possible ways to draw this map. I know that the B to VG distance, I know that distance is 14 because we calculated that on page 1. I now know that the B to CN distance, the body color to eye color distance, if this is CN, the distance from B to CN is 18, because I just calculated it. So does this map, is this a possible correct map based on what we know? Yeah. If this map is right, and I'm not saying it is, but if it's right, what would be approximately the distance from CN to BG?
4.
Okay, right. Because we know this distance is 18. We also know this is definitely 14 because we already calculated. So this is the only one we don't know for sure, but it would have to be 4. But how do we, what if it's this? What if the B gene is in the middle? Yeah. What if the B gene is in the middle and CN is, say, here? Because I know that distance is 14. No, I'm sorry. Let's do this. Let's make that VG. Sorry. I know that distance is 14. And I know the B to CN is 18. Is that also possible? We know this distance is 14 from page 1. We know this distance is 18 because we just calculated it. So yeah, this is equally valid based on what we know. But if this second map is right, then what would have to be the CN to VG distance? Not 4, but 32? So one of those has got to be right. One of those maps is the correct map. But we need more data. And luckily for us, we have more data on the next page because we have some data right here that will tell us the distance between the CN gene and the VG gene. And you know how to do it. Add up the total. 2,200 this time. Add up your recombinant types, which works out to be 88. Divided by 2,200 is 4. 4% or 4 map units, which conveniently enough matches one of the possible distances between VG and CN. Now we know this map was correct and this map is not. Because the distance from CN to VG is 4. It is not 32. So now I just draw that same map, the correct map, one more time. And let's see, here's B to VG with CN like this. Now, there's still things I don't know. Like, there's no way to tell from this data the exact locations of any of these genes. Like, this B gene might be way here on the tip, on the end. You know, so they all could be skewed down to the left. I don't know where they are left or right. I just know the order. I know that CN is further away from B than VG is. And I know this distance is 14. And I know this distance is 18. And let me show you one more thing real quick. Let's suppose I told you to draw the map. And on your paper, you wrote is that correct?
Yeah.
It doesn't look like what I drew, but is it correct?
Yeah.
Yes, because that chromatid can be flipped either way. If I pick this up and flip it, I'm going to get that. It's the same chromatid. It's just I flipped it. So as long as you have VG in the middle, and as long as you have the relative distances correct, it's right. It's the correct map. What we're going to do tomorrow is we're going to take that same map. We know that map is right. And we're going to add one more gene to it. I'm going to give you some more data. And we're going to add a 4th gene and we're going to draw one map that shows all 4 genes on the same chromosome. And if you can handle that, you've got this technique down pat. Any questions? Okay, we'll pack up and stand on an X. Hope you enjoyed your M&Ms today.